Okay, good afternoon, everybody, and welcome back. We are slowly going through our discussion about DNA. I'm deliberately taking it slowly and repeating things because this is so important. It's so important that you leave this class with some understanding about how DNA is structured, about how it works, and then about how it is used in forensic science. This information can inform so many things about that you're hearing at the moment. It can inform all your everything you're hearing about the COVID crisis, general biology, etc., as well as our specific interest in forensics. On Friday, today being Wednesday, on Friday there will be a live class at 4 o'clock. And that is to give you an opportunity to ask questions. It's a week or two since we had a live class. A few people ever attend the live class, usually no one. I presume that this means that nobody has any questions to ask, but I doubt that that is true. Friday is an opportunity for you to do so. Okay, especially about this section about DNA, which may leave some of you a bit lost, especially if you don't have any background in other sciences. Okay, so yes, DNA chapter 16, the title is The Indispensable Forensic Science Tool. It is indeed the indispensable forensic science tool, as long as it is there to be used. Not every crime scene is going to have DNA evidence present in such quantities as will be useful, but nonetheless, very frequently DNA evidence has proved to be crucial in solving of cases and bringing them to conclusion in court. And as I've emphasized before, one of the very important aspects about the use of DNA as a forensic tool is that its use is solidly scientifically based. It is based on very good experimental scientific evidence. And when we use it, we can place bounds of error around our use. An expert can stand up and say, give a range of likelihood. They can say it is 1 in a trillion, it is 1 in 300 chance, etc., etc. That is not the case for much of the other forensic evidence that we have talked about, which tends to be much more subjective. Nonetheless, I also want to emphasize that even DNA processing and analysis is subject to error. That's the first thing. And second of all, it is subject to malfeasance, as both of those things have happened and have impacted on cases where it has been shown that either DNA evidence was blatantly manipulated to implicate somebody, or where DNA evidence was so poorly analyzed that in fact the defense team could demolish the prosecution argument in using it, despite the fact, or perhaps because of the fact, that DNA provides such strong scientific evidence. It is extremely important that its handling, its analysis be extremely rigorous, and that all avenues of error are closed as far as possible. Okay, so I'm going to go over again some of the structure of DNA and move on also a little bit to tell you more about how DNA actually replicates itself. This becomes significant for us in forensic science because we're going to use a technique which manipulates the ability of DNA to replicate itself. And so here's our double helix. This should be quite familiar to you by now. The double helix is composed of DNA, is composed of a sugar phosphate backbone. There are two molecules which wind together to form the double helix. If we follow just one of those strands, we follow the sequence, we see that there's that the DNA is composed of sets of nucleotides. Here's one here, here's one here, there's one here. Each nucleotide is a monomer, and the monomers are joined together to form this extremely long polymer. Now, in human beings, we have in each cell, we have 46 chromosomes. Each chromosome, in actual fact, is one extremely long DNA molecule, extremely long, can be up to a meter long. That inside the nucleus is carefully packaged and wound to fit into that very small space. We have 46 chromosomes. 23 of those chromosomes are derived from your mother, and 23 of them are derived from your father. This becomes very important later on, as you'll see. You are inheriting genetic information from your mother, and you're inheriting genetic information from your father. And the genetic information from each can be different. All of your chromosomes are going to end up carrying the same genes. You're going to need a protein that you use in respiration, for example, but it may differ slightly. The maternal one may differ slightly from the paternal one, but they'll do the same thing. They'll have the same function. The function of DNA lies in the sequence of the nucleotides. For any gene, on one of the strands, there is information. And that information is contained in the form of these nucleotides in the sequence of the nucleotides. Here we have G, T, C, A, G, etc. This is not a random sequence. If this is the strand which is coding in the gene, what it is coding for is protein. And it codes for protein in words that are made up of 3 letters. So 3 of these nucleotides together will code for 1 amino acid. And proteins are made of amino acids. So this is how DNA actually works. It has words which specify an amino acid. And those amino acids are going to be put together in the order that the words come in the gene. How that is done, I'll refer to in a minute. So this sequence of nucleotides here is really, really important because that is the recipe, that's the code that the DNA contains. But the DNA is a double molecule. And it has a matching strand. Here's the matching strand here. If we have G, T, C, A here, we have to have a matching strand where the nucleotides read C, A, G, T, C. Because G must always match to C. Guanine always matches to cytosine. Adenine always matches to thymine. So if we get an adenine on this strand, we must have a thymine on that strand. Got a thymine on this strand, there must be an adenine on that strand. Cytosine on this strand, guanine on that strand. Cytosine this strand, guanine that strand, etc., etc., etc. This is really critical for the cell because every time that, let me just say, in addition here, this is just a simple short segment of DNA here to show you the same matching, right? The same base matching here. Here are this is a nucleotide. There's its phosphate. Here's another nucleotide. It's sugar bound to this phosphate. This one bound to that phosphate. This one bound to that phosphate. Very strong backbone. Across the middle here, holding the 2 strands together, weak hydrogen bonds between the bases there. You'll see the significance of this in a minute as well. Strong, strong, weak. But there are many, 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 many of those weak bonds. And DNA is in fact extremely stable. It's a very, very strong, stable molecule because of its bonding structure. Okay, so just picture this, that if I was to come along and I just wiped out that strand there, I just took it away completely. Could you recreate it? Yes, you could, very easily, because you know T must match to A, G must match to C, C must match to G, A must match to T. If I take the strand away, you'll very easily be able to tell me that the sequence on the strand is A, C, G, T. A, C, G, T, because they match those bases. Same as this. If I took away this strand completely and I asked you or the cell to recreate the matching strand, it could do it very easily, because A matches to T, C matches to G, G matches to C, T matches to A. So that these strands are talking to one another. If I separate them one from the other, I can recreate the other strand very, very easily. This is extremely useful for the cell. Just one thing that I will point out: the strands are said to run anti-parallel. We can look at them and see that they have their ends are different. Okay, have a look here. You'll see this here. It has that OH group at the end, and this end here has a phosphate group there. The matching strand is upside down. There's its OH end, and there's its phosphate end. They are said to be anti-parallel. So for esoteric reasons, that becomes important. We're not going to talk about it, but it has an impact on the functioning of the DNA. Okay, so every time a cell divides, it has to hand off a complete copy of its DNA across to the matching cell. Now just think about our DNA for a minute. I've already told you we have 46 chromosomes. Each chromosome consists of a very long single DNA molecule. So we've got 46 of these immensely long DNA strands inside our nucleus. And whenever our cells divide, all of that DNA has got to be replicated. It's all got to be doubled up so that each cell inherits a complete copy of the genome so that it can carry on functioning properly. That's point number one. Point number two, every cell in your body, except for the gametes that you produce, every cell in your body has the identical DNA and the full complement of DNA as well. So with very few exceptions, the cells in our body each contain a complete copy of our genome. Half of it derived from your mother, half of it derived from your father. And every time one of those cells divides, that genome has got to be reproduced entirely so that each cell can carry on healthily, doing whatever that cell is destined to do. And its fate is destined by what genes are active in the DNA for that particular cell. So the cell has to have some way of doing this, not only very, very accurately, but also very quickly. I'll point out, for example, that the cells of your mucous membranes divide probably a couple of times a day. The cells of your skin reproduce very, very rapidly as well, in a week. So they've probably doubled up several times. Other cells don't. Other cells never divide, or divide very seldom, like nerve cells. But many of our cells are constantly dividing. And every time they divide, they've got to double up their DNA really accurately. Then the structure that I've given you already, what you already understand about the structure of DNA, tells you exactly how DNA is going to double itself. Because what the cell is going to do is it's simply going to split these two. Here for our double strand, right? And this strand matches that strand, and that strand matches that strand. In other words, I'll phrase it another way. I'll say that this strand, in a way, is coding for that strand. and that strand is coding for that strand. Because it contains the recipe for that strand. This strand contains the recipe for that strand because they match T to A, C to G. So what is going to happen is the cell is going to separate the two strands from one another, and the cell is then going to recreate the missing strand. And it can do that because of those matching rules. It can do so. Not only that, it can do so very, very rapidly, and it can do so with great accuracy. In actual fact, in doing this, the cell is able to proofread what it's doing. The cell will check the whole time to make sure that it's put in the correct bases. If ever it makes a mistake, which does happen, but if ever it makes a mistake, it backs up, chews out the wrong base, and puts in the correct one in. So it's a very, very accurate process, and it takes place rapidly because all the functions of the cell have to stop when it's busy dividing. Its genes can't operate while it's busy dividing. So the division takes place very rapidly so the cell can get back to its normal functioning. So it takes place accurately and it takes place very rapidly. And that is thanks to the structure of the DNA and the fact that it matches, that it has these matching bases. So here is a strand, a double helix, which is busy reproducing. It's busy replicating. And the cell opens it up. It does it with enzymes. It's a very, very carefully controlled process. We won't go into all of the enzymes and everything. I'll just tell you that it's opening up the strand and it's running in this direction there. It's running that way there. And the diagram that we're looking at. So it's opening it up, splitting the strands apart. Now what is happening? All these strands are single-stranded now. They have a sequence, T, G, A, T here, and then the matching strand is A, C, A, T to match these here. So when you split these apart and they're single, it's very, very easy for the cell to bring in free nucleotides. The cell is full of nucleotides. We make them the whole time as we need them. We manufacture them or we get them from food, if in some cases. But the cell is full of these nucleotides, and using the correct enzymes, these nucleotides are brought in, and they're brought in to match the single nucleotide here. So here's a T, bring in an A. Here's a G, we bring in a C and join it to the A. Here's a T, bring it in, join it. Here's an A, bring it in. This is what is done on both strands. There's a little bit of a difference on how it's done on the two strands because they're anti-parallel, but don't worry about it for the moment. Don't worry about it. Just accept what I'm telling you. That in actual fact, that sounds so terrible, but in this case, I just don't want to get us into the weeds of exactly what enzymes are involved in and everything. So just take it from me that the cell is very easily able to match up these single bases with free nucleotides and recreate the strand, recreate another strand there. So now what we end up with, we had a single strand here. Now we end up with double strand again. And one half, we've got these two strands. And we look at each of them and we see that one half, one molecule, one half of it is old strand. And then one half is new strand that has been built modeled on the old strand. It's a very simple system. Of course, it's not that simple. It is complicated because it has to be very carefully controlled. But there are many enzymes involved. One of their really important functions, apart from building these strands, these new strands of DNA is to check that they're doing it correctly and to remove as much error as possible. That's, by the way, called the replication fork, this part where it's busy opening up. Okay, so there's one thing that I do need to tell you. There's one complication which we have to get straight in our minds and clear, because it rears its head again when we talk about one of the most important forensic techniques that are used to identify and type DNA. And have a look here. I'll just tell you that this replication in real life, it can start in many places in the human genome. It can start in many, many places in the chromosome and run in both directions. So this here is the replication, is a replication fork. We've opened up the DNA, and here's a replication fork, and it's opening up in that direction and replicating the DNA behind it. But here on the other side of this kind of bubble that has opened up in the DNA, it's going to carry on as well. It's going to replicate in that direction as well. And eventually all the bubbles just run into one another and you end up with two complete strands. I hope that that's clear. The diagram kind of makes it clear. Just remember, two sets of replication are taking place, one in that direction, one in that direction. And the thing that we need to understand is that in actual fact, replication cannot start straight away just with DNA. I can open up the DNA strands like this, but replication won't begin until a very short piece of RNA, ribonucleic acid, has been laid down. So in other words, we just forget about all the rest of this. Just have a look here at this part here. We've opened it up. We want to reproduce that section there where the arrow's running, right? We can't do that. With the enzymes just will not start working until I've put down a short length of ribonucleic acid. And ribonucleic acid, you'll remember, is single-stranded nucleic acid. And it uses uracil instead of thymine. So there's a couple of small differences. But here it becomes critically important because DNA replication will not start until we put down this RNA primer. Now, the RNA primer matches the bases, the DNA bases. If this was A, T, G, C, okay, this A, T, A, that'd be T, this would be A, this would be C, this would be G. In other words, we still obey the same rules, A to T, C to G. So it still obeys the same rules, but it makes this little patch of RNA stuck to the DNA, and that is called the primer. This is very useful for us. This becomes extremely useful because it means that we can identify an area of DNA that we want to reproduce, that we want to replicate. Later on, you'll see how we do it. But if we know what this sequence is here, we can make an RNA primer that could stick to that artificially and start reproducing this bit if we wanted to. We'll refer to this again later. For the moment, just think about the natural situation. This is the natural situation. When we want to replicate DNA, we open up the DNA strands, separate them, and we put down RNA primer. Once the RNA primer is down, then the cell can begin to replicate the rest of the DNA. You see this here, it says DNA pol 3. All that means is DNA polymerase. And a polymerase is an enzyme which makes DNA. A DNA polymerase makes DNA. So this is just a DNA polymerase, and it's running in this direction here, and it's making this new strand. It starts at the RNA primer and it just continues. And once this is started like this, it can continue for thousands and thousands and thousands and thousands of bases, for a very long time. Here it is here, a little bit later on in the process. Here's our RNA primer. Here's all of the new DNA strand doubling up on the old DNA strand that has been opened there. I know I'm belaboring the point, but this becomes critical to our understanding of a technique later on where we artificially increase the concentration of specific areas of DNA. And this is a special technique called the polymerase chain reaction, which we'll discuss in much more detail, probably in the next class. But in that technique, what we do is we would make an artificial primer and we'll put it in a solution of the DNA so that it will stick on a specific area. And we then put in all of the enzymes and everything, and we get the DNA to double up for us. We get it a specific area of DNA to be doubled for our purposes. Normally in the cell, this is what happens whenever DNA reproduces. The whole DNA strand, the whole chromosome gets reproduced. When we start to manipulate, we're going to see we're only going to be reproducing bits of the DNA. Okay, so just for your summary, here it is. Open up these two DNA strands. I'm going to lay down a little short primer. You bring in this enzyme called DNA polymerase, and you have available all of the nucleotides and everything that you need. Bingo. The enzymes will take care of the rest and rebuild your double strands for you. You'll end up with two new double strands. So just for exercise sake, I'm not going to shout them out, but please have a look at your, print these out and have a look at them and be ready, like in a test, whatever, be ready to behave like DNA polymerase. Okay, so you're the DNA polymerase. We've separated these two strands and we've got two different strands and we want now to recreate. And what are the bases that we put down here, all the way along there, and what are the bases that we put down there. So just write those out and it becomes very clear to you, because when you look at these two, you'll realize that what you've just done is you've just replicated part of a DNA molecule. Okay, so I do also want to just have very briefly talk about the function of DNA, the normal functioning of DNA in the cell. I've already told you that DNA contains the recipe for all of the proteins that are required for the functioning of our cells, and there are thousands and thousands of proteins. All the functioning of our cell is driven and directed by proteins, quite apart from the fact that much of the structure of our cell is also protein. And all of that protein is coded for in DNA. It's an amazing thing. Just think about the math of it. First of all, in DNA, there are only 4 nucleotides. There are 4 monomers that make up the whole of the DNA. Those 4 monomers code, in actual fact, for 20 amino acids. Proteins are also polymers, and their monomers are called amino acids. And there are only 20 amino acids that make up all of our proteins. But their order is very, very important, because the order determines 2 things. It determines, first of all, the structure, the 3-dimensional structure of the protein, and the order of the amino acids determines the functioning of the protein. And the functions of the proteins, there are just a gazillion different functions for the ways in which proteins can function, all determined by their structure and by the sequence of amino acids. So when we look at the DNA, we see that 3 bases in sequence will code for one amino acid. So in actual fact, when we work it out, if we've got 4 bases, we've got 64 possible combinations of 3. That's the threesome is referred to as a triplet, a DNA triplet. A DNA triplet codes for one amino acid. So for example, the triplet guanine, adenine, guanine, one after the other, codes for this amino acid called glutamine. CGT, cytosine, guanine, thymine, that codes for another amino acid called alanine, and so on and so on. The sequence is occurring is determined by evolution, right? These have all arisen in evolutionary time, all the sequences for all the different proteins. So unfortunately, well, I don't want to say unfortunately because it complicates our understanding, not unfortunately for the cell, but the DNA can't directly produce protein. We don't produce protein directly in the nucleus from DNA. Instead, there's an intermediary set of steps. And the first step is the DNA, the gene, the sequence of the gene is actually copied. And it is copied into the molecule you have already heard about, called RNA. Messenger RNA, in actual fact, is a copy of the genetic sequence of a gene. And that messenger RNA is exported from the nucleus to the cytoplasm of the cell. And there are these very small little bodies inside the cytoplasm called the ribosomes. And the ribosomes are able to latch onto the messenger RNA, and they will read the messenger RNA and they will translate it. And they translate the messenger RNA sequence into actual protein. When RNA is made from the DNA, a triplet ends up with a corresponding threesome of RNA, and that is called a codon. The RNA threesome is called a codon. The DNA threesome is called a triplet. The RNA threesome, codon, and the ribosomes are able to interpret each codon one by one by one, and they will bring in the correct amino acid and join the correct amino acid in the correct sequence, one after the other, one after the other, one after the other, to create the whole protein molecule. And the molecule may be very, very large. Many times there are thousands and thousands of amino acids in a single protein. So the messenger RNA is a transcript, if you like, of the DNA that is translated by the ribosomes. Now, I mention this also because it's really important to us at the moment, because there were two of the COVID vaccines. You will hear them referred to as messenger RNA vaccines. What they contain, those vaccines contain messenger RNA, which codes for one of the proteins of the COVID virus. So what the vaccine is doing is it's being injected into you, and it is contributing messenger RNA to your cells. And your cells then translate that messenger RNA, and they produce that single viral protein. And your immune system sees that viral protein and knows this is foreign protein, and your immune system reacts against that and gives you immunity. If ever you're exposed to the virus, hopefully your immune system recognizes the presence of the virus. The advantage of doing that is that messenger RNA, DNA, of course, is extremely long-lived. It lasts your whole lifetime. But messenger RNA is very short-lived. It's a matter of minutes to hours. its lifespan before it is degraded and disappears. So using a vaccine based on messenger RNA is very advantageous because it's not going to go backwards and turn into DNA. It can't do that. So it's not going to change your genetics at all. It just temporarily gives your cells a recipe for this foreign protein. Your cells very temporarily produce that protein, and it's soon recognized by your immune system that then gives you immunity. So that's one use, really important use of messenger RNA, artificial use of messenger RNA. Under normal circumstances, messenger RNA is read off the gene, exported to the cytoplasm, and it's translated in the cytoplasm to protein. Okay, so just really to emphasize the importance of the sequence of DNA, and then of course the resulting sequence of messenger RNA. To point this out to you, here is a sequence of amino acids. Here they are. Here. These are amino acids which form the protein hemoglobin. And hemoglobin transports oxygen in the red blood corpuscles. This is just to show you the importance of that sequence, of the sequence of bases. It's critically important. And as I told you, it's evolutionarily determined to try and get to the best functioning protein that's possible. So here's normal hemoglobin, and it consists of a series of amino acids. These are the ones that we are particularly in. Just look at this little sequence here: proline, glutamate, and glutamate. That's the normal. If you replace, first of all, this glutamate here is coded for by the sequence GAG, by the triplet GAG. And that normally ends up producing glutamate. If we just change one base, just one nucleotide, we make this GTG, this no longer codes for glutamate. It now codes for another amino acid called valine. Here it is here. So now instead of proline, glutamate, we've now got proline, valine, glutamate. Well, that might look like it's a minor change, but it's actually huge, because this here is the hemoglobin that produces the disease of sickle cell anemia. So just from this one nucleotide change here, we change that amino acid. Just that one amino acid change ends up changing the whole functioning of the hemoglobin. The hemoglobin no longer works the way it is supposed to do, and it produces a disease syndrome called sickle cell anemia. That is the importance of the sequence. So in general, we expect DNA sequence to be extremely stable and not to contain errors like this. This kind of error we refer to as a mutation. Now, this mutation probably arose tens of thousands of years ago in some population of human beings and has been handed on, because that is what happens. We hand on our genetic sequence, we hand on our DNA sequence to our progeny in the form of our gametes. And if any of our DNA has become mutated like this, we run the risk of handing on that mutation to our progeny. Just the take home, though, is that all of our information contained in our DNA has an origin. It has its origin in our parents, in their parents, in our great-parents, and great-great-grandparents, and great-great-great-grandparents. It's all been inherited from these different sources. And that's something that we're going to need to remember as we start to talk about trying to identify an individual's DNA. Okay, so I'm going to finish off there. We'll discuss in more detail next time exactly how forensic scientists now address the problem of trying to type DNA and tie it to an individual. I'll just tell you before we finish that we're going to start talking about a section of DNA which is not actually gene. The genes code for something useful. They code for the proteins, but there are huge areas of our DNA which do not code for anything. Often the function of that DNA is unknown. We're not exactly sure what it is. Sometimes we do know that it has function and has regulatory function. But the take-home message is that in between the genes in our DNA, there are big stretches of DNA which contain other kinds of sequence. And one of the things that happens is that they begin, you will see areas where they contain short lengths of DNA that repeat after one another, one after the other, after the other, after the other. We see the same little DNA sequence, like bricks in a wall, and each brick identical to the previous brick. And then those, very often, the number that are contained in a specific area of DNA is very variable from person to person. So you inherit that number. One inherit those numbers from your parents, but they, don't forget, as we've just talked about, have come from an immensely long genetic line. And these areas are subject to change, which you can inherit. So these little areas of repeating DNA are called DNA tandem repeats. Tandem because they're next to one another. But you might have GCG, 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 GCG are one after the other, after the other, after the other. They don't have any genetic, they don't contribute to any protein production. They are just there, but you inherit them from your parents. Your parents inherit from a long line of inheritance. Okay, so we'll stop there and I'll discuss this in more detail next time.